Hey everyone, and welcome back to the channel. Today, we're diving deep into the very essence of what makes you, you. We're talking about the blueprint of life itself. DNA and RNA. Have you ever wondered how a single tiny cell knows how to grow into a complex organism, like a human, a tree, or a blue whale? The secret lies in two incredible molecules, and by the end of this video, you'll understand exactly how they pull it off. So, let's start with the basics. Imagine you're building something with Legos. You need individual bricks to construct anything, right? In the world of genetics, these bricks are called nucleotides. Every single strand of DNA and RNA is built from these tiny units. Each nucleotide has three parts. First, there's a sugar molecule. Think of this as the main body of the Lego brick. In DNA, this sugar is called deoxyribose, and in RNA, it's called ribose. It's a tiny difference, but it has huge consequences, which we'll get to later. The second part is a phosphate group. This is like the connector on the Lego brick. The phosphate group of one nucleotide links to the sugar of the next one, forming a long, strong chain. This is what we call the sugar-phosphate backbone, and it gives the DNA and RNA molecules their structure and stability. It's the scaffolding that holds everything together. Now for the most exciting part, the third component, the nitrogenous base. This is the part of the Lego brick that carries the actual information. There are 4 different types of bases in DNA: adenine, which we call a guanine, G, cytosine, C, and thymine, T. Think of these 4 letters as the alphabet of life. Every instruction, every trait, every characteristic is written using just these 4 letters. It's an incredibly efficient coding system. So, we have our building blocks, the nucleotides. How do they come together to form the famous DNA molecule? This is where it gets really beautiful. DNA isn't just a single string of nucleotides. It's a double helix. Picture a twisted ladder. The 2 long sides of the ladder are the sugar-phosphate backbones we just talked about. The rungs of the ladder are where the magic happens. Each rung is made of 2 bases, one from each side of the ladder, linked together in the middle. But they don't just pair up randomly. There's a very specific rule, known as the base pairing rule. Adenine or A always pairs with thymine, or T and cytosine, or C always pairs with guanine. Or G, you can remember this with a simple mnemonic: apples in the tree and cars in the garage. A with T, C with G, always. This pairing is not just a neat little detail. It's absolutely fundamental to how DNA works. This complementary pairing means that if you know the sequence of bases on one strand, you automatically know the sequence on the other. For example, if one strand reads a TCGC, the opposite strand must read TOCG. This is crucial for DNA's ability to replicate itself perfectly. When a cell divides, it needs to make an exact copy of its DNA for the new cell. The double helix unwinds, and each strand acts as a template to build a new complementary strand, resulting in 2 identical DNA molecules. It's a brilliant, self-replicating design. So we have this incredible double helix structure storing a vast amount of information. Your DNA contains about 3 billion of these base pairs. If you were to stretch out the DNA from a single human cell, it would be about 2 meters long. And yet, it's all packed into a cell nucleus that's microscopic. How's that possible? The DNA is supercoiled and wrapped tightly around proteins, like thread on a spool, allowing this immense library of information to fit into such a tiny space. This library, your genome, contains the instructions for building every protein your body needs to function. Proteins are the workhorses of the cell. They act as enzymes to speed up chemical reactions, provide structural support, transport substances, and so much more. The sequence of A's, T's, C's, and G's in your DNA determines the sequence of amino acids that make up each protein. A different sequence of bases results in a different protein, which in turn leads to different traits. like your eye color, your hair texture, or even your predisposition to certain diseases. Now, you might be thinking, okay, DNA is the master blueprint, the ultimate library of information stored safely in the cell's nucleus. But how does the cell actually use this information to build things? The library is valuable, but you need a way to read the books and carry the instructions to the construction site. This is where our second molecule, RNA, comes into play. RNA, or ribonucleic acid, is the indispensable messenger and factory worker of the molecular world. While DNA's primary job is long-term storage, RNA has a much more active hands-on role. Let's revisit the differences we touched on earlier. RNA is typically a single-stranded molecule, not a double helix like DNA. It uses the sugar ribose instead of deoxyribose. And there's one change in its cast of bases. Instead of thymine T, RNA uses a base called uracil, or USO in the RNA world. Adenine pairs with uracil. The process of using the DNA blueprint starts with something called transcription. Think of it like this. You go to the library, find the specific book or chapter you need, which is a gene on the DNA, and you make a copy of it. You don't take the original book out of the library because it's too valuable. In this cell, an enzyme reads a segment of DNA and creates a complementary messenger RNA or mRNA copy. This mRNA molecule is a short, portable transcript of the instructions needed to build one specific protein. Once the mRNA copy is made, it leaves the safety of the nucleus and travels out into the cytoplasm. It's the main part of the cell, where the protein-building machinery is located. This machinery is called the ribosome, and it's also partly made of another type of RNA, ribosomal RNA, or rRNA. The ribosome acts like a factory assembly line. It latches onto the mRNA molecule and starts reading its sequence of bases, 3 at a time. Each three-letter word, called a codon, specifies a particular amino acid, the building block of proteins. But how do the correct amino acids get to the ribosome factory? That's the job of a third type of RNA, transfer RNA, or tRNA. Each tRNA molecule is like a specialized delivery truck. One end of the tRNA recognizes a specific codon on the mRNA, and the other end carries the corresponding amino acid. As the ribosome moves along the mRNA, tRNA molecules arrive one by one, dropping off their amino acids in the exact order specified by the mRNA code. The ribosome then links these amino acids together, forming a long chain that folds into a functional protein. This whole process, from mRNA to protein, is called translation. So, to summarize the flow of information, DNA stores the permanent master plan in the nucleus. Through transcription, a temporary mRNA copy of a specific gene is created. This mRNA travels to the ribosome. Then, through translation, the ribosome reads the mRNA code, and with the help of tRNA, assembles a protein amino acid by amino acid. This is the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. It's an elegant and incredibly precise system. The stability of the DNA double helix makes it perfect for long-term, reliable information storage, protecting the genetic code from damage. The transient, single-stranded nature of RNA makes it ideal for its role as a temporary, disposable message. It's created when needed and degraded after its job is done, allowing the cell to control which proteins are being made at any given time. This division of labor is why nucleic acids are truly the blueprint of life. DNA is the architect's master plan, kept safe and secure. RNA is the set of working instructions taken to the construction site, used to build the structures, the proteins that make everything happen. Every living thing on Earth, from the simplest bacteria to the most complex animal, uses this fundamental system. It's the universal language of life, connecting us all. Thank you so much for joining me on this journey into the molecular heart of life. I hope you now have a clearer picture of how DNA and RNA work together to store and use genetic information. It's a process that's happening in trillions of your cells right this very second. If you found this video helpful and interesting, please give it a thumbs up and consider subscribing to the channel for more deep dives into the wonders of science. Let me know in the comments if you have any questions or what topic you'd like to see next. Thanks for watching, and I'll see you in the next video.